Hello and welcome to the session. In this session, we are going to see about the process of chromatography for separation of molecules. So, chromatography is a separation technique which is based upon the separation of molecules on their interaction with the mobile and the stationary phase. In the technique of chromatography, there is a stationary phase and a mobile phase. The mobile phase could be a liquid or a gas. So the molecules which are to be separated, they interact both with the stationary and the mobile phase. The mobile phase, as the name says, is the phase which can move or which can flow. So the molecules which are to be separated, they move along the flow of the mobile phase. So it depends upon the interaction of the molecules with the mobile phase and the stationary phase. If the molecules interact with the mobile phase more, so they are going to be carried away with that mobile phone, and therefore they travel greater distance. The molecules which interact with the stationary phase more, so they bind to the stationary phase and therefore they travel less distance. So based upon the relative affinity of the molecules with the stationary phase versus the mobile phase, a given type of molecules could be separated from another type of molecules. So the basis of separation is the interaction of the molecules with the mobile versus the stationary phase. And this in turn governs the distance of migration. And based upon the relative distance of migration, the molecules they could be separated from one another. This is a diagrammatic representation showing the stationary phase by means of this rectangle, and these are the two types of molecules shown in blue and yellow color. So the molecules they are allowed to move along the stationary phase, and this is arrow shows the direction of migration of the mobile phase. Now, when these molecules are allowed to move along with the mobile phase, here we see that the blue molecules they travel greater distance. This is because of the reason that the affinity of these molecules with the mobile phase is more as compared to their affinity with the stationary phase. So these molecules are going to be carried away along with the flow of the mobile phase, and therefore these molecules travel greater distance. The other molecules, represented by this yellow circle, they have greater affinity for the stationary phase as compared to their affinity for the mobile phase. So therefore, they are not going to be carried away to a similar extent along with the mobile phase. Since they are able to interact more with the stationary phase, so their migration is going to be less. In this manner, the molecules with greater affinity for the mobile phase, they will travel greater distance. And the molecules having less affinity for the mobile phase will travel less distance. In this manner, these two types of molecules could be separated from one another. To keep it simple, in this illustration, only two types of molecules are shown. But of course, in real life scenarios, there could be more than two types of molecules, or sometimes there could be thousands of molecules present in a given mixture. A limitation of the process is that if two molecules they are very similar or exactly identical in their properties, then their migrations are going to be the same. So they cannot be separated from one another. So this phenomenon of separation is based upon the difference in the properties of the molecules. Several different types of properties of molecules could be used for separation through chromatography. For example, proteins or other molecules, they could be separated from one another on the basis of their charge, on the basis of their size, hydrophobicity, or some other properties. So this is a table from the book, and it shows the different types of chromatographic techniques. Depending upon the mobile phase, there could be two types of chromatography in broader terms. If the mobile phase is a liquid, that is called as a liquid chromatography. Or if the mobile phase is a gas, then that is called as gas chromatography. So, regarding liquid chromatography, this could be further divided into two major groups. The planar chromatography, in this type of chromatography, the stationary phase is present in a two-dimensional plane. So, paper chromatography is one such type of chromatography which you have carried out in some of your other labs as well. And thin layer chromatography, it contains a thin layer of silica or embedded on a on an inert surface, and the molecules are allowed to move through it. So these are examples of planar chromatography where the stationary phase lies in a flat two-dimensional plane. As opposed to planar chromatography, in a column chromatography, the stationary phase is present in a cylindrical column, and the molecules to be separated are allowed to move through that column. One such example is ion exchange chromatography, in which the stationary phase, it contains charges on it, and the molecules, based upon their charge, they are able to interact with the stationary phase, and hence separation is based upon charges. If the charge of the stationary phase and the molecules to be separated is the same, then they cannot bind to the stationary phase because of charge-charge repulsion. However, if the charges are opposite, then the molecules of interest can bind to the stationary phase. So ion exchange chromatography is based upon the charge of molecules. Size exclusion chromatography is important for separation of molecules on the basis of their molecular mass. High performance liquid chromatography is done by using automated systems. The machines, they have pumps and a fluidic system in it, so they automatically take the sample and the buffers, and then they pump it through the column. During affinity chromatography, it is based upon the specific binding affinity of a molecules of a particular type to the stationary phase. For example, there are certain type of proteins which contain histidine residues on them. So the histidine residues, they can bind to nickel ions which are present on the stationary phase. So these proteins which contain histidines on them exposed on the surface, they can bind to the stationary phase containing nickel ions. So this is an example of a specific binding affinity. Similarly, several different types of enzymes, they could be purified by using a stationary phase which contains the substrate of that enzyme. So the enzyme is going to bind to the substrate which is present on the stationary phase, and other proteins will not be able to bind. So in this manner, we make use of specific binding affinity of a particular molecule which is present on the stationary phase to capture a given molecule from the mixture. And then gas chromatography is a type of chromatography in which the mobile phase is a gas. Generally, gas chromatography is used for either volatile substances, which could be easily converted into a gas phase, or molecules which have a small molecular mass. One important parameter for describing the rate of migration is called as the RF value. The RF, it is described by the expression, that is the distance traveled by a given molecule along the stationary phase, its ratio to the distance traveled by the mobile phase along the stationary phase. So this migration is a function of the distribution ratio. The distribution ratio is given by this expression, that the concentration of a biomolecule in a stationary phase, its ratio to the concentration of the molecule in the mobile phase. So that means the ability of a substance, of a molecule, to interact with the stationary phase versus its ability to interact with the mobile phase. If a substance is going to partition more in the stationary phase, it will travel less distance and hence the distribution constant will be more. On the other hand, if some molecules are having greater affinity for the mobile phase, then they will partition more in the mobile phase, will travel greater distance, and hence will have a low value for Kc. So one important question in this regard is that why there is a need to purify proteins. So chromatography could be used to purify proteins and other types of molecules as well. So why the purification of proteins is needed? Generally, the proteins we isolate are coming from cells because proteins are produced in a cell. So we lyse the cell, break open the cell, and the proteins are going to be released in a buffer. Typically, the cells contain thousands of different proteins in them, and the required protein is one of those thousands of different proteins. So, in order to study the biological activity of that protein, it has to be purified. Otherwise, there may be interacting substances or other interfering proteins which may be present in that mixture. Many other different types of molecules they could be interfering with the protein of interest as well, or they may be able to alter the protein of interest in some way. So in order to study the biological property, one important reason to study its property. Another important thing for which purification is needed is its is the requirement for the protein to be crystallized. If we want to know about the function of protein, the function is dictated by the structure of that protein. And in order to determine the structure, we have to crystallize that protein. So in order to crystallize the protein, a certain level of purification is needed. So that's another important thing. In order to study the structure or the biological function of the protein, it has to be purified. Another important reason is that many proteins they have specific applications. For example, many proteins like insulin or antibodies, they are therapeutic agents. So they have to be administered to human beings, and unless purified to a sufficient level, they cannot be administered to people. So in order to have this application, this is a picture from the book, and this is representing a purification strategy for proteins. So this is called the CIP purification strategy. C stands for capture, I for intermediate purification, and P for polishing. So initially, the proteins they are obtained from a given source. The source for obtaining proteins they are cells of a living thing because proteins are produced in cells. There are some other cell-free protein synthesis systems as well, but generally proteins are extracted from the tissues of an organism. It may be an animal, it may be plant, or it could be bacteria. So the cells of the organism they are lysed. After lysis, the proteins of the cells are released into a buffer, and the proteins are extracted, and then this lysate is then clarified. That is, we remove the insoluble materials from that lysate. So generally, separation is done by centrifugation. So the larger particles, unlysed cells, and debris are going to be separated. So we get a clear lysate. This process is called as clarification of the lysate. Now, this lysate is a mixture containing thousands of proteins which are obtained by cell lysis. The first step during the purification strategy is the capture step. In this process, the protein of interest is isolated, concentrated, and stabilized. Proteins, they could be concentrated by using salting out, in which we add salt, and therefore the proteins, they could be precipitated, and then they could be dissolved in the suitable buffer, and therefore that results in concentrating the proteins. Sometimes some proteins, they need cofactors or additional substances for their stability. So the idea for the initial step of purification is to isolate the protein and to stabilize it. The mixture still contains a lot of impurities in it. The next step is called intermediate purification, which is focusing on removing most bulk impurities and obtaining partially purified protein. And the last step is polishing, in which the most of the bulk impurities have been removed previously, so any other remaining impurities, they are removed in this process. So proteins, they could be purified by using chromatographic techniques on the basis of a variety of properties. One important property on which separation could be done is the charge of proteins. And in this process, the proteins are separated by using a stationary phase which contains charges on it. This is called as ion exchange chromatography. Another property for separation of proteins is the hydrophobicity of the proteins. So this type of chromatography is called hydrophobic interaction chromatography, in which the stationary phase is hydrophobic. So the proteins will bind to the stationary phase depending upon their hydrophobic behavior. The proteins which are more hydrophobic will bind to the stationary phase with a stronger affinity. Another important method to separate proteins is on the basis of their size. This type of chromatography is called as size exclusion chromatography. In this chromatography, the stationary phase contains pores in it, and the proteins, they move through the stationary phase on their ability to penetrate those pores of the stationary phase. Those proteins which are larger in size as compared to the pore size, they won't be able to migrate through the stationary phase. Whereas those proteins which have a smaller size than the pore size, they would be able to move through the stationary phase. So this type of chromatography separates molecules on the basis of their size. And the next type of chromatography is called as affinity chromatography, in which the molecules, the proteins, they could be separated from one another on their specific binding affinity to the stationary phase. Different types of affinity tags could be used for separation of these proteins. For example, a histidine tag. A histidine tag is added during the process of cloning to a recombinant protein. The proteins which contain a histidine tag on them, they show binding affinity to metal ions, typically nickel metal ion, which is bound to the stationary phase. So a stationary phase contains nickel ion, and the protein of interest, they contain a histidine tag on them, or simply called as a his tag. A his tag is a combination of, generally it is a combination of 6 histidine amino acids. These amino acids are added to the protein during the process of cloning, and the recombinant resulting protein is containing the his tag on it. So proteins bearing the his tag show high affinity to nickel ions which are present on the stationary phase. So these metal ions, the nickel ions, they are immobilized on the stationary phase, and the protein of interest show affinity and it binds to that stationary phase. So therefore, this type of chromatography is called as immobilized metal ion affinity chromatography. That is, the metal ions, they are immobilized onto the stationary phase, and the proteins containing the his tag have a binding affinity to that stationary phase. Other proteins which do not have the his tag on them, they won't be able to bind to this column. Hence, in this manner, only the protein which contains the his tag on it it will bind to the stationary phase and therefore could be separated. Other proteins will not bind. Another important type of tag is the maltose binding protein. It is a protein which binds to carbohydrate maltose, and so if this tag MBP is actually a protein, so this protein could be appended to the protein of interest, and this could be appended through the process of gene cloning. The vector which is used for cloning of the protein, it contains the gene for maltose binding protein, and the gene of interest is cloned along with this protein. So this protein is going to be attached to the protein of interest and will be covalently bound. So the maltose binding protein has a binding affinity with dextrins, with carbohydrates containing maltose residues on them. So if the stationary phase contains maltose residues, then this protein, along with the protein of interest, is going to bind to the column. Other proteins which do not bind to carbohydrates, they will be eliminated from that stationary phase, and only this required recombinant protein would be able to bind to the stationary phase. So this is again, these two are examples of affinity chromatography where the protein of interest has a specific binding affinity for the stationary phase. So this slide shows the fundamental steps of the process of chromatography. The first step is equilibration of the column. The resin is a stationary phase, which is generally made up of small particles, and these particles are packed in form of a cylindrical column. So the process of equilibration, it allows the stationary phase to be in conditions which allow the protein of interest to bind to it. For example, the protein of interest may be stable at a given concentration of salts, or it may be stable at a given pH. So the process of equilibration is done to make the conditions of the stationary phase suitable for the binding of the protein of interest. Equilibration is done in the following manner, that we allow a buffer having the proper conditions for the binding of the protein to move through that stationary phase, to move through that column. So when that buffer, it flows through the column, so the conditions of the column become compatible for the binding of the protein. For example, if a protein is able to bind to a particular stationary phase at a pH of 8, so we pass a buffer of pH 8 from the column. In this manner, the stationary phase, it become equilibrated so as it can bind to the protein of interest. So equilibration is done by passing a given buffer through the stationary phase. The next step is application of the sample. So the sample after initial processing is then added to the column, and the protein of interest is going to bind to the column. Those proteins which do not bind to the column, they will be removed during the washing step. Sometimes it happens that only one protein can bind to the column, but this is only possible for affinity chromatography. For other types of chromatography, many different proteins can bind to the stationary phase in addition to the required protein. Those proteins which are not bound by the stationary phase or the column, they are removed through the process of washing. Washing is done by using a buffer. So the wash buffer is passed through the column, and those substances, those molecules which are not bound to the column, they are going to be removed away. And only those molecules are going to stay which have a binding affinity with the column stationary phase. After washing is done, then the molecules of interest, they are bound to the column. Later, we change the conditions. Again, the conditions are changed by passing a suitable buffer through the column, and by changing the conditions, the proteins are going to be eluted from the column. Sometimes we can alter the ionic strength of the buffer. In other types, we can change the pH of the buffer. Or sometimes we can add a competing substance which can displace the bound proteins from the column. The process of removing the proteins from the column and releasing them into the buffer is called as elution. So different proteins, they will be eluted under different conditions. In this manner, we periodically alter the conditions, and proteins with different properties are going to be eluted at different conditions. The proteins which are eluted in a particular condition, they are collected in a separate tube. So in this manner, the proteins are going to be fractionated. They are going to be separated from one another. Later, after the proteins have been eluted, then the column is regenerated. Regeneration means to prepare the column, to remove any other substances which are remaining in the column, and to prepare it for a next run. If an immediate run is not needed, then the column could be stored in a suitable solution. The columns, they are not kept dried, rather they are stored in a liquid. Generally, during storage, we add 20% ethanol so as to prevent any microbial growth in the column. So one important type of chromatography we are going to talk about now is ion exchange chromatography. Ion exchange chromatography separates molecules on the basis of their charge. It is important to know that charge is dependent upon pH. Proteins, they contain many different types of ionizable groups on them. For example, carboxyl groups, amino groups, hydroxyl groups, and many other functional groups which have dissociable protons on them. So, depending upon the pH, the ionization of these groups is affected, and in turn, this affects the charge on the proteins. So, we adjust the pH of the buffer in such a manner that the protein of interest contains a charge on it, and now it would be able to interact with the stationary phase through the charge-charge interaction. In this type of chromatography, the stationary phase, it contains charge groups on it. The stationary phase, it may contain positively charged groups or negatively charged groups on it. The proteins having an opposite charge, they can bind to the column. And the proteins with similar charges will not be able to bind to the column, and they would be removed away. So if the stationary phase is positively charged, then negatively charged proteins can bind to that column. And if the stationary phase is positively charged, then negatively charged proteins can bind to it. Once the protein of interest, they are bound to the stationary phase, then elution could be done by adding competing ions. Say for example, the stationary phase is positive and negatively charged proteins bind to the column. So later we can displace the bound proteins by adding more negative ions. These competing ions generally are provided by increasing the salt concentration, say sodium chloride. So the chloride ions, when their concentration increase, they are going to displace the proteins which are bound to the stationary phase. There are two types of ion exchange chromatography: cation exchange chromatography and anion exchange chromatography. This is an illustration which shows the process of ion exchange chromatography. This blue circle here represents the stationary phase, and this stationary phase is positively charged. So, during the equilibration step, we allow the buffer to pass through the stationary phase. The negatively charged ions in the buffer, they are going to interact with the stationary phase and they will be bound by the stationary phase. So these small blue circles here, they represent the buffer ions or the salt ions present in that buffer. Since the stationary phase is positive, so these negative charges present in the buffer are going to interact with the stationary phase and are going to be bound here. In this illustration, only these few charge salt ions are shown here. Of course, they will be bound on the entire surface of the stationary phase. The stationary phase generally is made up of small beads which contain functional groups on them, and the functional groups are charged. So this represents one bead of the stationary phase. To simplify the illustration, the binding is shown only on this part of the bead, but actually the bead contains the charge groups all over its surface, and binding will take place all over the surface of this bead. But to simplify, it is only shown in this part of the stationary phase. So when we equilibrate, we allow a buffer with the suitable conditions to pass through the stationary phase. So the buffer ions, which are oppositely charged, they are going to bind to the stationary phase. In this case, the negatively charged ions of the buffer will be bound here. Later, once equilibration is completed, then we add the sample. The sample here is a mixture of three proteins, but in actual conditions, it may be containing many thousands of proteins in it. So this blue circle is again representing the buffer ions because the proteins are dissolved in a buffer, and this green star is a positively charged proteins, and the other two proteins represented by the triangle and the square, they are negatively charged proteins. So when these proteins are added, the positively charged protein has the same charge as that of the stationary phase. So this protein is not able to bind to the stationary phase, so it will be removed along with the flow of the buffer. In this case, the buffer is the mobile phase. So that protein which bears the same charge as that of the stationary phase, it will be repelled away and it will not be able to bind to the stationary phase. Now these two proteins, they both are negatively charged, so they both will be able to bind to the stationary phase. The binding of these proteins is shown in a sequential manner in this illustration, but actually all proteins which have negative charges on them, they would be able to bind to the stationary phase simultaneously. Here in this illustration, the protein represented by the triangle, it has less negative charge and its binding affinity to the stationary phase is low, as opposed to the binding affinity of this protein with the stationary phase. Now we add the buffer and then we wash the stationary phase. So those proteins which are unable to bind to the column, they will be removed during the washing step. Those proteins which are bound to the column are going to stay there. After washing, then we are going to elute the proteins from the column. In order to do so, we increase the salt concentration. Now we allow a buffer to pass through the column which contains a relatively high concentration of salt in it. So these are the negatively charged ions which are present in the buffer. So as the concentration of these negatively charged ions is increased, they are going to displace the bound proteins. The protein having low binding affinity with a stationary phase, it will be displaced from the column at a low concentration of competing ions. So therefore, as the salt concentration is increased, the negatively charged ions are going to displace the protein with low affinity first, and it will be removed away. Now, this position will be occupied by the negatively charged salt ions. The other protein, represented by the square, has higher binding affinity with a stationary phase, so it cannot be displaced at a low salt concentration. Then we allow a buffer with a higher salt concentration to pass through the stationary phase. Now, this is represented by these circles here, which show the negative charge ions. Now, as the salt concentration increase, now these salt ions, they are going to displace the high affinity protein from the stationary phase, and in this manner they occupy these positions on the stationary phase. So here in this illustration you can see that during the washing step the positively charged protein was removed from the column. At low salt concentration the protein with low affinity was eluted, and then at high salt concentration the protein with high affinity was eluted. Now when this buffer is going to flow through the column, it is collected in separate tubes or containers. So previously all of these proteins were present in the same mixture, but later when they are eluted from the column, they are collected in different tubes or containers. In this way, the proteins are going to be separated from one another. They are going to be distributed in various fractions. So sometimes it is called as fractionation of proteins. This is a diagrammatic representation showing the stationary phase which contains positive charges on it. So negatively charged proteins can come and bind here, and later these proteins are going to be displaced by the negative ions of the salt. Many a times the salt used in the process is sodium chloride. So the chloride ions, as their concentration increase, they are going to displace this negatively charged protein and they are going to bind to the stationary phase. Now in this type of chromatography, the stationary phase is positive and negative charge protein binds to it, and then it is exchanged by negatively charged salt ions. Since in this process, negative charges are being exchanged on the stationary phase, so this is called as anion exchange chromatography. In the other type of ion exchange chromatography, the stationary phase is negatively charged. So a positively charged protein can come and bind here, and later when the salt concentration increase, the sodium ions are going to compete for binding and they are going to displace this protein from the column. So in this manner, the stationary phase is negatively charged and positive charges bind to it, and later these positively charged proteins are going to be exchanged with the positively charged salt ions. So in this type of chromatography, positive charges are going to be exchanged on the stationary phase. So this is called as cation exchange chromatography. pH is an important factor in governing the charges on the protein. Pi, or in other words the isoelectric point, is representing the pH at which the net charge on the protein would be 0. That means isoelectric point is corresponding to a pH where the number of positive charges on a protein becomes equal to the number of negative charges. Since the number of positive charges is equal to the number of negative charges, so the net charge on the protein would be 0. So if the pH is equal to the isoelectric point of the protein will have no net charge. Its net charge would be 0, hence it will not be able to bind to the ion exchange column. If the pH of the buffer is more than the isoelectric point, the protein will have a net negative charge on it and it will bind to an anion exchange column. If the pH of the buffer is less than the isoelectric point, then the protein will have a net positive charge on it and it would be able to bind to a cation exchange column. So that means it is important to know about the isoelectric point of a protein, and the pH is adjusted in such a manner that the protein of interest would be able to bind to a suitable ion exchange chromatography column. So it is an important consideration regarding ion exchange chromatography. These are the different types of ion exchangers which are used in ion exchange chromatography. This shows the functional groups which are present on the stationary phase. The beads of the stationary phase, they could be made up of agarose or polyacrylamide. Agarose is a carbohydrate and polyacrylamide is also a polymer. So the stationary phase could be either agarose or it may be made up of polyacrylamide, and this stationary phase contains charge groups on it. One type of charge group is quaternary ammonium. Quaternary ammonium is a strong positive charge. The other groups include diethyl aminoethyl, which is a weakly positive charge stationary phase. Another one is diethyl aminopropyl, which is also weakly positively charged. So these are showing the anion exchange functional groups. The cation exchangers, they include sulfopropyl, which is a strong negatively charged functional group. Others include methyl sulfonate, which is also a strong negatively charged functional group. And another one which is commonly found is carboxymethyl, which is also a negatively charged group, but it is weakly negative charge. So different types of ion exchanger matrices, they could be used for separation of a given protein. So this figure shows the process of ion exchange chromatography. Initially, the sample is applied to the chromatography column. The column contains the stationary phase in it. The stationary phase, as we have talked about it earlier, could be made up of agarose, or sometimes it could be made up of polyacrylamide containing the positive or negatively charged functional groups on it. So the ion exchange medium, the stationary phase, is packed inside the column, and then the sample is added to the column. So the proteins are now added to this column, and along with the flow of the buffer through the column, the proteins are going to move through the stationary phase. The proteins which are able to bind to the stationary phase, they're going to stay in the column, while those proteins which are unable to bind to the column, they are going to move through this, and eventually they are going to be removed from the column along with the flow of the buffer. So these proteins which are removed from the column, they will be collected in separate tubes. Now the proteins which are bound to the stationary phase, they could be separated from one another by gradually increasing the salt concentration. So first we add a buffer through to the column which contains a low salt concentration in it. So the proteins which are having low binding affinity to the stationary phase, they will be removed by using a low salt concentration, and they could be collected in these different tubes. Then later we increase the salt concentration and we allow this buffer to pass through the column. So now the proteins with intermediate binding affinity will be displaced from the column, and they will be removed from the column along with the flow of the buffer, and therefore could be collected in separate tubes. And then we further increase the salt concentration in the buffer, and we add that buffer to the stationary phase. Now the proteins which have high binding affinity to the column, they will be removed from the column, and these proteins are now going to be collected in these different fractions. This is a graphical representation. It shows protein concentration on the y-axis and the fractions of the protein on the x-axis. So here, this represents the point where the buffer with low salt was added. So gradually, this protein shown in red color was eluted from the column. So this protein has a low binding affinity, so it was eluted at low salt concentration. Then the salt concentration was increased, and the proteins with intermediate binding affinity will be removed from the column. And when the salt concentration is even higher, the proteins with the highest binding affinity will now be removed from the column. So in this manner, the proteins, they could be collected in different tubes or in different containers, and hence they could be separated from one another. This illustration shows the manual mechanism for adding the different salt concentrations to the stationary phase and removal of the proteins from the column. These days, there are now automated machines which automatically take up the protein solution from a sample reservoir, and they take the buffers from the buffer reservoir, they mix the salt solution with the buffer, and they automatically inject those salt concentrations into the column. And then the proteins which are removed from the column, they are monitored by using a built-in UV lamp. Proteins show a typical absorbance at 280 nanometer. So when the proteins are removed and they are going to be removed from the column, so close to the column is located a UV lamp. So when the buffer is passing through that detector, the absorbance is low. But when a protein is eluted and now that protein moves through the detector, so it shows increase in absorbance. So in this manner, we can detect when a given protein is going to be eluted from the column by monitoring the absorbance at 280 nanometer. This is a typical chromatogram. A chromatogram is a graphical representation of the elution pattern of proteins. Here, on the x-axis is the volume of the buffer, and on the y-axis is the salt concentration. These blue peaks are representing protein concentration in terms of their absorbance at 280 nanometer. So initially, the buffer was passed through the column, and that was meant for equilibration. Later, the protein sample was injected. Now, when protein was injected, the proteins which were not able to bind to the column, they will be removed from the column as such, because they are unable to bind to the stationary phase. So this increase in absorbance here represents those proteins which were not able to bind to the column, and they were removed from it. The proteins which were able to interact with the column, they would stay in it. Now, those bound proteins could be removed by increasing the salt concentration. This red line here represents the increase in salt concentration. So as the salt concentration is increased, we see another peak here, which represent a low affinity protein to be eluted from the column. This protein was collected in a separate collection tube. Then the salt concentration increases further. Now another protein with relatively greater binding affinity will be removed from here. As the salt concentration increases further, the proteins with even higher binding affinity will be released from the stationary phase and they will be eluted from the column. And eventually, the salt concentration was increased to the highest level, in this case, which was 1 molar. So the protein with highest binding affinity, they would be removed from the column as well. So all these proteins, they were collected in different fraction in different collection tubes in form of fractions, and therefore they could be separated from one another. This is an SDS page which shows the proteins which are eluted from the column. There could be several ways to analyze the proteins which are eluted from the column. One of the commonly used method is electrophoresis of proteins, which is SDS page or sodium dodecyl sulfate polyacrylamide gel electrophoresis. So it's a type of electrophoresis in which we denature the proteins and we run them on the gel. So this is the required protein. So this was the sample which was loaded onto the ion exchange column. So you can see each of the bands represent different types of proteins. And then in this, you can see this is a mixture of proteins. And this was the required protein. And then the various fractions, they were collected from the ion exchange column. And you can see that in these fractions, the protein of interest has been purified and other proteins have been removed from these fractions. As compared to this mixture, these fractions, they have a relatively purified protein. Other ways to detect protein would be to monitor the biological activity of that protein or to detect that protein by using an antibody through a Western blot. For example, if a given protein is an enzyme, then we can collect the fractions from the column and can look for the biological activity, for the enzyme activity in those fractions. So the fractions which contain the required enzyme activity, they will be containing the required protein of interest. So the next type, when there are like phases, for example, we have two polar phases. For example, salt and water, they both are polar. So they can easily interact with one another and salt can be dissolved in water. Similarly, if we have both non-polar phases like oil and hexane, they both are non-polar and hence they can interact with one another. So if we have like phases, they would be able to interact with one another. But if we have unlike phases, then these unlike phases will not be able to interact with each other. For example, if you put oil in water, this represents, the blue circles represent water molecules and the yellow circles represent oil molecules. Now these oil molecules are present in water. Oil is non-polar. On the other hand, water molecules are polar. So water molecules will not be able to interact with the oil molecules because water molecules are polar but oil is non-polar. So this would result in repulsive forces between the polar and the non-polar molecules. As a result of which, these non-polar molecules or hydrophobic molecules, they will be forced to interact with one another. And of course, eventually they will merge with one another and form a larger droplet of oil. So this illustration is an example of hydrophobic interaction. So when we have unlike phases, there are hydrophobic substances or non-polar substances present in a polar environment. The non-polar substances are unable to interact with water molecules, so they are forced to interact with one another. This is called as hydrophobic interaction. That is, interaction is taking place between hydrophobic groups or hydrophobic species, and that interaction is taking place because they are present in an aqueous phase, and the aqueous phase is polar. So when non-polar substances or non-polar groups are present in a polar environment, they will be forced to interact with one another because they are going to be repelled uh, by the polar solvent around them. So this interaction between the non-polar substances is called as hydrophobic interaction. Proteins could also be separated by using a hydrophobic interaction. In this case, the stationary phase is hydrophobic, and the hydrophobic parts of a protein would be able to bind to the stationary phase, and proteins could be separated on the basis of their hydrophobicity. Greater is the hydrophobicity of a protein, higher would be its binding affinity to the stationary phase. Proteins contain several different types of hydrophobic amino acids. Certain amino acids, they have aromatic groups on them. Other amino acids, they have aliphatic hydrocarbons on them. So these aliphatic hydrocarbons or aromatic hydrocarbon side chains, they are non-polar groups. So proteins, they have a distribution of both polar and non-polar groups on them. Now proteins are present in an aqueous solution. So in most proteins, their hydrophobic regions are not exposed because of the same phenomenon. The hydrophobic regions of a protein are generally buried in the core of the protein where they do not come in contact with water. So in order to separate proteins on the basis of their hydrophobic regions, it won't be possible if the protein is folded in its original structure. Because when the proteins are folded, most of their hydrophobic amino acids are buried in the core of the protein, and the hydrophilic amino acids are present on the surface of the protein. So we can open up the structure of the protein and can expose the hydrophobic regions by addition of salt. Generally, the salt of interest for this purpose is ammonium sulfate, and the concentration of salt is generally kept high, around 1 to 2 molar. This phenomenon operates in the following manner. As the decrease, the protein will fold back into its original structure, and the hydrophobic regions will again be shielded. So the proteins, when we want the proteins to bind to the stationary phase, we increase the salt concentration in the protein solution. The protein structure opens up. And then when we have to elute the protein from the column, we decrease the salt concentration, and when the salt concentration decreases, the protein folds back into its original structure, and hence the hydrophobic regions become shielded. When they become shielded, they won't be able to interact with the stationary phase. Hence, that protein will be eluted from the column. So that was the basis of separation through hydrophobic interaction chromatography. Now, this is a chromatogram representing the elution pattern of different proteins from a hydrophobic interaction column. On the x-axis is the order of the proteins in which they are eluted. On this y-axis is the absorbance, representing the amount of the protein when it is going to be eluted from the column. And on the secondary y-axis is the salt concentration, represented by red line. These are the structures of these 4 proteins, and the yellow circles, they represent the hydrophobic amino acids present in a given protein. So you can see that the hydrophobic content of the amino acids is increasing here from left to right. So initially the salt concentration is high, so all of the proteins, their hydrophobic regions are going to be exposed, so they all would be able to bind to the stationary phase. Now, when the salt concentration decreases, the proteins are going to fold back into their original structure and their exposed hydrophobic regions will again become shielded. And when the hydrophobic regions become shielded, they cannot interact with the stationary phase. And hence, that particular protein will be released from the column. So here we see that the protein with the least hydrophobic content is going to be eluted first, and then as salt concentration decrease further, now a relatively higher hydrophobic protein will be eluted, and as the salt concentration is increased, the more hydrophobic proteins are going to be eluted from the column. In this manner, the proteins, they could be separated on the basis of their hydrophobic character. The proteins with low hydrophobicity are going to be eluted first, and proteins with higher hydrophobicity will be eluted later. This is an STS page of a protein showing the purification of protein through two steps of chromatography. So here, this is a lane of the gel which shows this is a protein of interest, and then there are many other bands of proteins as well. So it's a complex mixture containing many proteins in it. And first, the protein was separated through anion exchange chromatography, and the fraction eluted from anion exchange chromatography was then subjected to hydrophobic interaction chromatography. So here you see that in this lane marked as 4, if you would compare it to 1, so you can see that the other unwanted proteins, they have been removed from this required protein. Which separate proteins on the basis of their molecular mass. So this technique separates the proteins on the basis of their size and their shape. In this type of chromatography, the stationary phase is made up of a porous material, and it has pores and channels in it. The proteins, which have a size smaller than the pore size, they go inside the beads of the stationary phase, and therefore they travel greater distance through the column. Other proteins which are larger, they will not be able to go in these pores, and therefore they take less time to travel through the column. This is diagrammatically represented here. So the column, the stationary phase, is made up of beads which contain pores in them. So this is a cross section, and you can see that these black structures are the beads, and inside the beads there are pores and channels. Now the proteins which are smaller, which are of a smaller size, they will be able to go into the channels of the bead. And when the protein will be released from this bead, again it will encounter another bead and can go into the channels of that bead. Then the protein moves further, and then it will be trapped in the channels of the next bead. And eventually it will go into the pores of the next bead and the next one, and in this manner the small proteins will travel through these beads, through the channels which are present in these beads, and then eventually they will be released from the column. Larger proteins, on the other hand, will not be able to penetrate inside these pores because their size is greater. So therefore, larger proteins will be able to move through the spaces between the beads, and hence they travel less distance and they are going to be eluted first. This is also represented in this part of the illustration. So we see that this green color thing is the stationary phase, and we have proteins of 3 different sizes here. So the protein having the smallest size is going to penetrate into the pores of the bead and is going to stay here, and then it will migrate to the next channel or the pore of the bead, and it will take it longer time to travel and eventually migrate from the column. The proteins with intermediate size, they will penetrate the beads, but will penetrate only to a partial extent, and hence they will take relatively more time to be eluted. They will take relatively less time to be eluted from the column. Whereas the proteins with the largest molecular mass, they will not be able to penetrate the beads, and hence they are going to move through the interstitial spaces. So the protein with larger size are going to be eluted from the column first, followed by smaller proteins. Because smaller proteins are going to spend more time traveling through the pores of the beads, and hence they will be eluted later through the column. Larger proteins, they spend less time in the column because they are unable to go into the channels of the beads, and they are going to move through the interstitial spaces, and hence they will be eluted first from the column. In this manner, the proteins are going to be separated on the basis of their molecular mass. Depending upon their ability to penetrate through the beads of the column. There are different types of chromatography beads available with different types of pore sizes. So depending upon the type of the protein which is to be separated, we can select a suitable type of the stationary phase having suitable pore size. So the larger proteins, they will travel through the interstitial spaces mostly, and their penetration within the beads would be very less, so therefore they are going to be eluted earlier. Smaller proteins, they will move through the channels of the beads, hence they stay in the column for a longer duration of time, and the smaller molecules, they will be eluted later from the column. And then by using a similar graph, we can determine the molecular mass of the protein of interest. So another important implication of size exclusion chromatography is that it could be used to determine the molecular mass of a protein. In addition, it could also be used to determine the oligomeric state of a protein. Some proteins, they have only one polypeptide in them. They are called as monomeric proteins. That is, they have only one polypeptide. Other proteins, they may be oligomeric. That is, they have more than one polypeptide in them. For example, hemoglobin is tetrameric. It means it contains 4 polypeptide chains in it. Some proteins are dimeric. Others could be trimeric, that is having 2 or 3 subunits in them. Some proteins can have even more subunits in them. So when the subunits, they oligomerize, they give rise to a functional protein. Size exclusion chromatography can also be used to study the oligomerization of a protein. So we can carry out chromatography under the native conditions when the protein is in its original structure, and can find the elution volume of that protein, and hence can determine the mass. Then another run of the same protein could be done under conditions which dissociate the subunits from one another. This is called as a denaturing condition, in which the subunits are dissociated from one another. For example, we can add a detergent to dissociate the protein subunits from one another. And then we carry out size exclusion chromatography again. If the size of the protein, if the elution pattern of the protein remains the same, that means the protein is not dissociated into any constituent components. So the protein is made up of only one subunit. However, if we see several different peaks of the protein eluted at different molecular masses, so that means the protein of interest was made up of several subunits which have dissociated from one another, and now each subunit is going to be eluted corresponding to its own molecular mass. In this manner, size exclusion chromatography can be used to determine the molecular mass and also to find the types of the monomers which are present in a given protein. Also, this could be used to determine the oligomerization state of a protein. For example, if we run a protein under its original native form, the protein is eluted at a volume which corresponds to 80 kilodalton. But when we add a detergent to the protein solution and run that protein on size exclusion chromatography column, now the protein elutes at a molecular mass of 20 kilodalton. That means this protein is a tetrameric protein. It contains 4 subunits of 20 kilodalton. So when the 4 subunits are assembled with one another, so the protein has a mass of 80 kilodalton, which is the original native form of the protein. But when we add the detergent, the subunits are dissociated, and all of the subunits, they are of 20 kilodalton each. So we observe one peak of 20 kilodalton on the size exclusion chromatogram. In this manner, this method of chromatography could be used to determine the molecular mass and the oligomerization state of a given protein. So next we move on to the next type of purification through chromatography, which is affinity chromatography. Affinity chromatography is done for the proteins which have specific binding affinity for a given stationary phase. Many times we can add tags to the proteins through molecular cloning, and by using those tags we can purify the proteins by using affinity chromatography. Here we are going to discuss only one such type of. This is called as immobilized metalline affinity chromatography. So here, this is the stationary phase. The stationary phase is made up of agar beads or agarose beads, and it contains a functional group on it. And associated with these functional groups are the nickel ions. So the nickel ions, they are immobilized onto this stationary phase. Now, when proteins are added and the proteins contain histidine on them, a histidine tag on it, then the histidine tag proteins are going to bind to the stationary phase by means of this nickel ion coordination complex. The stationary phase groups are shown in red color here, and these blue groups are representing the histidine tag which is attached to the protein. The histidine molecules, this is the structure of the amino acid histidine. The histidine molecule, it contains the side chain. This is called as the imidazolium group. The imidazolium group contains these nitrogens, and by means of these nitrogens, these histidines, they can form a coordination complex with the nickel ions. In this manner, only the proteins which contain exposed histidines on them, they would be able to bind to these nickel ions through coordination. Other proteins which do not have exposed histidines on them, they will not be able to bind to this stationary phase. So in this manner, the histidine tag proteins will have a specific binding affinity to this nickel column, and they will bind to it. Later, these bound proteins, they could be eluted from the column by using a competing agent called imidazolium. This is the structure of imidazolium. This is the structure of imidazole, and it resembles the imidazolium ring of histidine, having a similar nitrogen configuration. So when imidazole molecules are added, imidazole molecules, they compete with the hist tag proteins, and they displace the protein, and they themselves bind to the nickel ions present on the column. So in this manner, histidine tagged proteins, they will have a specific binding affinity to the nickel ions on the stationary phase, and they are bound here. Other proteins, they will not bind to the column and they will be removed. And later, when we add imidazole, it is going to displace the protein and itself will become associated with the stationary phase. One important significance of affinity purification is that only the required protein which has high binding affinity to the stationary phase can bind to it. All other proteins, they will not be bound to the stationary phase. And then by using a suitable competing agent, the protein of interest could be released from the column and hence could be collected. So in this manner, affinity purification increases the yield of the protein, and it also increases the efficacy of the process because it only requires one step of chromatography to get the required protein with a greater yield and uh, with a greater yield and high efficacy. This is a diagrammatic representation to show the process. So the protein of interest here in this case is produced by host cells, and the host cells in this case are E. coli cells. So the bacterial cells, they contain a plasmid, and the plasmid has the recombinant gene on it with the histidine tag. So the cells are allowed to produce the protein of interest, and then the cells, they are they are lysed to release the protein, and then the protein of interest is released in the buffer. So the lysate is centrifuged to remove the debris, and hence it is added to the nickel containing column. The protein of interest will be bound to the nickel stationary phase, and then it is washed to remove the unbound proteins, and later the required protein could be eluted from the column by using imidazole. In this manner, all other impurities could be removed in a single step of chromatography. So affinity chromatography is quite useful because it results in producing high yields and with greater efficacy of purification.